All right. Well, hello and welcome to PanKind's research webinar. I think we'll get started. My name is Michelle Stewart and I'm PanKind's CEO. So thank you so much for joining us today on World Pancreatic Cancer Day. Just a little bit of housekeeping before we start. I need to let you know that the information that's presented in these webinars is of a general nature and should not be considered personal or medical advice. Always seek independent advice relevant to your specific situation. The opinions of the presenters are their own and do not necessarily reflect the views of PanKind. Also, if you experience technical difficulties during the webinar, please let us know via the Q&A chat bar, which you can see on your screen, on the right of the screen, or email sophia@pankind.org.au. A recording of the webinar will be available from our website, pankind.org.au, and only those presenting on screen will be in the recording. During this webinar, attendees' mics and cameras will be off. So thank you very much to everyone for joining. So welcome. Over the last couple of years, PanKind have taken the opportunity to do a research update on World Pancreatic Cancer Day. And today is a day of global action and awareness to shine the spotlight on pancreatic cancer, on the disease. So organisations and people affected by the disease are raising their voices today to spread the awareness, and we're proud to join with these groups. World Pancreatic Cancer Day is organised by the Global World Pancreatic Cancer Coalition, and we're really pleased that Jess Abelson from Purple Our World in Australia is one of the leaders of the committee for World Pancreatic Cancer Day. So thank you to Jess. I'm not sure if people have seen a lot of pancreatic cancer activity on social media today, or whether you've been able to wear purple in your workplace or do things to raise awareness of the disease. But I have been seeing, where I sit, a lot of activity, which has been fantastic. So today we are going to be hearing from Professor Ross McKinnon about the pancreatic cancer research that PanKind has been funding, but also the pancreatic cancer research landscape, both in Australia and globally. Before I hand it over to Ross, I just wanted to give a little bit of context to PanKind so that everybody can understand what we're trying to achieve with the research that we are funding. So we're an organisation based in Australia, based in Sydney, but with volunteers and ambassadors located all around the country. We are committed to raising funds to invest into research to improve survival for people infected by pancreatic cancer. We really represent those who are affected by the disease, and we believe that no one has a more vested interest in seeing change happen. As a result, the funds raised by PanKind and our community are so important because there's such a passionate focus behind it. Those who donate are either trying to donate to support someone that they love or to ensure that nobody else has to go through the same situation in the future. And as a result, we take this responsibility very seriously. We need to invest these donations so that we receive, so that they go into the most promising projects that will achieve the outcomes that we are looking for. We are looking to improve survival, as I say, and we've got a very laser focus on doing that. Now, a lot of organisations might do everything in funding research, fund from all over the spectrum, and that is something that we have definitely done previously. But this year we did an evaluation. We wanted to make sure we could be as effective as possible. So we looked at the entire research environment to work out how we could make a difference to those who are affected by the disease. We came up with two areas of focus. We know that people who are diagnosed earlier have a much higher probability of better outcomes. And this is mainly because they're either eligible for surgery or existing treatments might be more effective. We know that only 10% of people are diagnosed at this point in time. And so where we could see would make the most significant difference was to be putting focus and funding on making sure that people are diagnosed earlier in the future. And Ross will be talking a little bit about some of the work we're doing here. Secondly, as I mentioned before, we know that 90% of patients are not responding currently to current treatments very well. And so we will continue to support research into new treatments of the disease. The next thing I'll say is we know that time is very important for our supporters. So for those people who support us, they wish that there was treatments yesterday. And so as a result, we put a really big lens on the time frame that it will take for the research that we fund to reach patients. And so we use this and we evaluate the research projects with this context in mind. So just to let you know, AVNA Foundation, or as it was previously known, or PanKind, has now invested $9.2 million into research, with 1.6 of that being invested this year. So it's been a big year for research. We also will be investing a significant amount in December this year. So make sure you stay tuned to the new projects that we'll be announcing at that point in time. The reason that we were able to invest such a large amount of money in December is due to the success of the Remember September campaign. And I know that a number of people involved on the call today were involved in Remember September, and you raised $1.5 million for pancreatic cancer research, which is an incredible milestone and something that I think took us all by surprise. So I think that the funds invested, I know that the funds that are invested from that campaign will actually make a huge difference to people affected by the disease. And the grants are currently in review, but we will be announcing some pretty important projects shortly. The other thing I just wanted to let you know is that research is really competitive across all the funding schemes. A lot of researchers are funded by the NH and MRC, which is a National Health and Medical Research funding scheme. Currently, only 10% of people who apply for grant funding in this scheme are successful, which is difficult. But I mean, it would be a really difficult job with all the amazing researchers that are out there and all the worthy diseases that they could be funding. So what can we do as an organisation to try to help that our researchers in pancreatic cancer? One of the most important things that a charity or groups like ours can do is to provide that early stage funding to researchers when they need it, so that they can get that data, that proof of concept data, that then make them much more competitive to the overall research funding schemes. So this is really where we are focusing our research dollars, and Ross again will talk about that shortly. So what does success look like for us? So success looks like we want to improve survival to 30% by 2030. So this is quite a technical measurement. So we want to make sure that by 2030, 30%, as opposed to the 11% are currently surviving for 5 years. What does this really mean? What we want to say is when someone hears of a diagnosis of pancreatic cancer, that people say, that's the one they can treat, isn't it? So we believe that would be an outcome of success. In the shorter term, if we know that the researchers that we are funding then go on to receive bigger grants, funding from bigger grant schemes, that is also a success. So at that point, I'd like to introduce the chair of our scientific advisory panel, Professor Ross McKinnon, to give us an update. Ross is Strategic Professor in Cancer Research and Matthew Flinders Distinguished Professor of College of Medicine and Public Health down in Flinders. Ross is one of the most influential, I would say, researchers in the country, and we are so fortunate to have had his guidance in pancreatic cancer funding over the last many years. He's provided oversight to the foundation and has managed a very eminent panel of clinicians and researchers, some of the top in the world, and has been instrumental to help boosting pancreatic cancer funding and capacity in Australia. So at that point, I will hand over to Ross to jump into it. And I think Ross is on mute.
Hello, Michelle.
Oh, hi. Thanks, Ross.
I do apologise. I actually had a blackout, which was interesting, but I've managed to retrieve things. Thank you for the opportunity to talk a little bit about the work that the Scientific Advisory Panel looks over each year. what I thought I would do, obviously it's very difficult to cover the whole topic of pancreatic cancer research in a half-hour session. So what I thought I would do is talk a bit about the types of grants that we award at PanKind, to talk about some of the successful applicants and what they're actually doing, but also to try and talk about, well, to contextualize it for the international sort of pancreatic cancer community. I'm just going to share my screen. There we are. I think everyone can see that. So first of all, let me introduce the scientific panel. They are all highly distinguished clinical individuals. We have two Americans. We have Andrew Lowy, who's from San Diego, University of California, San Diego, and also Eileen O'Reilly from the University of California, San Francisco. Then we have Lorraine Chantrell, one of the most distinguished pancreatic cancer physicians in Australia. John Chen, who is a pancreatic surgeon from Flinders University in Adelaide, and Chris Bagley, who would be known to many as the former Chief Medical Officer of Australia. And Chris is also a survivor of pancreatic cancer, and so he serves that consumer role as well, and brings a fantastic amount of experience to the panel. Sorry, I'm just, yep. So, as Michelle has already said, we have awarded over 9.2 million in pancreatic cancer research funding across some of the best institutions across the country. And I've got a photo of each of the successful applicants over the various years, and I apologise if there's anyone that I have forgotten. As we can see, it's a real mix of individuals. It's a mix of clinicians, a mix of scientists, a mixture of early stage and later stage career people. And I'll be talking about the work that some of these people are doing as we go along. Well, we have essentially over the years, we've really had four main funding schemes. The early detection scheme is one that Michelle has already alluded to. It's a strategic area for PanKind, and of course it talks to the issue of the need for early diagnosis. And of course, the fact that most individuals or many individuals diagnosed with pancreatic cancer have spread of disease, and that of course affects prognosis. So we had a deliberate scheme for early detection, but we also have had a number of early detection grants in some of the other schemes over the years, and I'll talk about one of those. Our innovation grants are smaller grants. These are, as Michelle was indicating, they're often small amounts of funding for projects to begin projects or to change direction with projects. We believe they're critically important, and I'm going to talk about one of those and highlight how that has led to success in achieving further funding. Our Accelerator Grant scheme is a much larger funding scheme in terms of the size of the actual grants. And this tends to talk to major areas of research and major collaborative groups, often that are quite well established, but are seeking funding for major programs of work. And I'll touch on a couple of those and in particular try and put those into an international context. And then the last scheme is the collaboration scheme, which is one that we've introduced recently, and that's designed to try and bring groups together to tackle particular issues that are relevant to pancreatic cancer and of course to advance the research. Over the years, we've identified that most of our grants fall into four key research areas, and we've used different wording for these at different times, but today I'm going to refer to them as early detection, understanding the cancer microenvironment, which is really about understanding the biology of the actual cancer, the resistance to current therapeutic modalities, and also novel therapeutics and repurposing existing agents. We see all of these as critically important. As I said, we've used early detection as a strategic scheme this year, but we see the other three as being just as important. Sorry, I'm just having a bit of trouble advancing my slides. Okay. Just before I start on the PanKind funded research, I thought it was worth mentioning that you can go to the Australian New Zealand Clinical Trials Registry, enter the term pancreatic cancer, and it identifies 203 current clinical trials in pancreatic cancer. I've just listed on this slide and the next one 6 of the trials. I haven't listed these for any particular purpose other than just to try and reflect the breadth of the research that's being undertaken. For those who don't know what the registry is, it's basically somewhere where we lodge information about clinical trials. It assists with recruitment. It assists with other researchers identifying what is happening. There are a number of recognised registries around the world. The Australian New Zealand one is, of course, the one that we use, but we also use clinicaltrials.gov, the US-based one. These are really important registries. They do give you real insight into what is happening in the space. And I'll talk about some trials that will be listed on some of these registries as we go along. And as I said, there is a second slide. We can see the breadth. Some of them are obviously treatment-based. Some of them are more around services and so forth. So let's start with early detection grants. And I thought I'd give you a couple of examples from the recent round that we had. They're somewhat similar in one way. The first one of these is led by Rachel Neal from QIMR, Queensland Institute of Medical Research, Berghofer Medical Research Institute in Brisbane. This project focuses on detecting pancreatic cancer earlier before symptoms, and it's essentially a data linkage project. So it's trying to identify through data individuals that we wouldn't, sorry, if I just take a step back, general population screening is not feasible due to the low incidence of pancreatic cancer in the population. And so we need to focus on screening strategies on those that are more likely to develop pancreatic cancer and create a surveillance program for those people. This program is about identifying these people through data linkage. We think it's a very exciting project. And it's a project that we hope will lead to the identification of a population of individuals that will be followed closely. The second example I wanted to give you was from a project being led by Andrew Metz from the Epworth Healthcare Centre at the Royal Melbourne. And this project, actually, I should say the Epworth in association with the Royal Melbourne. This project centres on the development of a novel clinical screening tool in people with diabetes and chronic pancreatitis, and it focuses in particular on one group of people. These are people diagnosed with diabetes after the age of 50, and they're diagnosed with a type of diabetes we call type 3C diabetes. It's actually a poorly understood form of diabetes. It's poorly understood by many health professionals, but also by the broader community. But it certainly seems like these are individuals who have diabetes as a result of pancreatic damage. There's a numbers of ways that pancreatic damage can occur. These particular researchers have assembled a very strong clinical cohort, and they'll be trying to identify from that group things that are likely to predict those individuals that are most likely to develop pancreatic cancer. So both of these approaches, as you can see, are really around identifying individuals that are most at risk, and they try to overcome the fact that broader screening is somewhat difficult in terms of the low incidence of pancreatic cancer and the cost that would be involved. But we'll talk a bit more about that in a moment. Again, I'm having some difficulties here. I do apologize. We had a power blackout and it's not recovering very well. What I might just do is switch my screen. Let's see how we go there. If not, is it possible for you, Sophia, to control my slides?
I can try to help you out with that, Russ, if it doesn't work in a sec, for sure.
Yeah, I'm just not having any luck progressing them. So let's have a look here. Okay, so as we've already identified, early detection is a major interest of PanKind, but I also wanted to contextualize what's happening around the world in the early detection space. And I'd just alert you to PanCAN for those who are not aware. I'm sure most of you would be aware of it. It's the Pancreatic Cancer Action Network based in the US. They are investing a significant amount of money in early detection, and the one I've highlighted here is their early detection initiative. This is very similar in some ways to what we've already talked about. It's about identifying those individuals that are likely to be most at risk, but on perhaps a much larger scale. The other thing that's happening around the world is the development of blood-based biomarker tests, and there are two listed here, and I'm sure many people have read about these. Grail, the company is called Grail, and the test is called Galleria. This is a multi-cancer detection test that's based on a single blood sample, and it's essentially a pan-cancer test. So it's about identifying multiple types of cancer, but included amongst those are pancreatic adenocarcinoma and also pancreatic neuroendocrine tumors. It's a circulating DNA-based technology. I'll talk a little bit more about that in a moment. And it's really analyzing DNA for tumor-specific mutation profiles and methylation patterns. The second test, Immunovia, the test is called IMRAY, PanCAN-D. It's for early detection of pancreatic cancer in individuals that are at high risk, and it's based on 9 blood-based biomarkers, so effectively a signature. And there's a number of clinical trials involved at the moment that are utilizing this particular test. So just taking a step back and talking a little bit about these early detection tests. Essentially, they involve what we call liquid biopsies, and liquid biopsies is basically trying to obtain information that's going to help us at various stages of the management of cancer in terms of early detection, but also in terms of surveillance for micrometastatic disease, and also for looking at treatment selection and response to treatment and things like this. The key to these liquid biopsies is that blood contains many things that we think are going to be, and we know are going to be very useful in terms of supplying us with biomarkers for these various things that we want to do. And the examples of the sort of things we find in these liquid biopsies are circulating DNA, as I've already said, circulating tumor cells. We also find exosomes. Exosomes are small lipid particles, or lipid ringed particles, if you like, that essentially carry a payload of information. And that information includes RNA, protein, a whole host of things that we would find very useful from the perspective of cancer research, cancer diagnosis, cancer management, and so forth. So we've already seen these two tests that are based on the liquid biopsy, and we have a number of research proposals that come through the advisory panel that also focus on this liquid biopsy. It's something that I work a lot with in my own research. We're very interested in where I work in terms of the role of liquid biopsies in assessing early response to drug treatment and also resistance to drug treatment. Very importantly, with those tests at the moment are in the stage of trying to obtain regulatory approval. They are actually available. They're not subsidized in any way. They must be paid for. Is that you, Sophia?
Sorry, it seems to have uploaded itself. One
it
second.
Does. So it looks like it's going to respond to me now. I just have to share a different screen. So. Okay, if we can go back.
Here you go, Russ. You should be able to control it from here now.
Okay. So, okay, thanks. Right. So importantly with these tests, they are at the stage, as I said, of obtaining regulatory approval. This is just a little schematic of what happens with diagnostic tests such as this. And of course, we've seen a number of things over the years, and a good example is the Oncotype, which is a microarray-based test for breast cancer, and gives us a feel for patients most likely respond to certain treatment strategies. They all must go through this process of accumulating, obviously in the initial stages, analytical validity and clinical validity, and showing that they're going to be clinically useful. Then, of course, seeking regulatory approval, and of course, reimbursement, and in Australia that is of course through federal government means, and then of course incorporation into clinical workflow, and each of these steps is associated with challenges. So the tests that we're seeing emerge around the world look exciting in many ways, but there's still a lot of work to do in terms of trying to optimize them and ensure we have the correct evidence base for appropriate introduction into clinical practice. If I now turn my attention to the pancreatic cancer grants supported by PanKind again and talk about innovation grants, do you remember these are relatively small grants that try to fund perhaps a new project or a change in direction of project? And the example I've got here is actually, and I'm having trouble reading my slide there. Okay. So this innovation grant was led by Dr. Ying Zhu from the University of New South Wales. And this is a really interesting grant that's based on what we've been talking about, a liquid biopsy. But this particular grant combines nanoengineering with exosome analysis. You remember exosomes are those particles that we talked about, fragments that are released by cancer cells. Exosomes are actually something where there's been a massive amount of literature emerging over recent years. We've learned a lot about what they are for, and they are involved in cellular communication and so forth, and transporting materials between cells. But as I said, they carry a cargo of molecules that we can use in various ways in terms of cancer diagnosis, cancer monitoring, and hopefully early detection. So this work by Dr. Zhu involves essentially the development of a nanoengineering sort of device. This device will act at the point of care. Blood samples can be taken, analyzed using this device, and hopefully identifying a signature of biomarkers on exosomes that can guide us in terms of early detection. It's quite early work in terms of where it's at, but it's exactly the type of work we try to fund with our innovation grants. The second example of innovation grant is one by a fellow called Professor Shane Gray. And actually, if you can see there, the title of 2 grants, that's because Shane was successful in successful in obtaining 2 innovation grants, one in 2017 and one in 2019. And Shane's work is highly innovative, and essentially it focuses on a checkpoint. We call these immune checkpoints, and these are essentially a safety mechanism that we have in our immune system. But unfortunately, in the case of pancreatic cancer, some of these checkpoints seem to be involved in resistance to immune-based therapies. Shane has identified a new checkpoint, and he's also identified a molecule involved in that checkpoint, and he's recognized with his work that mutations in that particular molecule can influence the immune response to various treatments. So his work involves essentially genetic manipulation of that target to try and maximize the immune response we see in pancreatic cancer. It's very exciting work, and in really good news, and we can just see here, Shane has actually provided me with this slide, and I thank him for that. Shane is based at the Garvan Institute. Shane was just in the most recent round of the NHMRC, and Michelle has already talked about the fact that there's about a 10% success rate, so it's incredibly difficult to get success in these new NHMRC rounds. I'll touch on that at the very end of my presentation. But Shane has received a ideas grant. Ideas grants are targeted at innovative ideas. It's work that has evolved directly from the 2 innovation grants he received from PanKind. It is about, as I said, this immune response. It's about this checkpoint and the ability to perhaps ramp that immune response up or down. So essentially a sensor, if you like. And he's used a sort of a volume switch to indicate what he's trying to do here. It's very, as I said, innovative work, very exciting. Congratulations to Shane, and also congratulations to PanKind for funding the early work that has led to this funding success. The next thing I want to talk about were collaboration grants, and as the name would imply, these are larger grants. Our collaboration grants, as I said, is a relatively new scheme, and I've got a number of examples of the collaboration grants here. The first one is a collaborative led by Ashley Pohl, Dr. Ashley Pohl from the Olivia Newton-John Cancer Research Institute at La Trobe. As we can see from the photo there's a number of other individuals involved, quite senior individuals and clinicians as well. And as we can also see, Ashley is a relatively early stage career researcher, and it's wonderful that she was successful in obtaining this grant, leading this grant. As the name suggests there, this grant focuses on the reprogramming of the tumor environment, microenvironment, to improve response to therapy. And in particular, it involves a molecule called hematopoietic cell kinase. This is a protein that is found in a type of immune cell called macrophages. And their team has shown that cells that have elevated levels of this particular kinase, and the kinase being a type of enzyme, these particular cells that have high levels of this particular kinase are better at supporting tumor growth. And on the other side is when they downregulate this particular enzyme, they find that the cells lose their ability to promote tumor growth across various cancer models. So this is essentially a basic science research with very good translational potential. And it's a really exciting piece of work. And again, congratulations to Ashley for receiving such a prestigious collaborative grant at a relatively early stage of her career. The second example of a collaborative grant I wanted to highlight was one that's being led by Professor Frederick Hollande. He is based at the University of Melbourne, as we can see. And we also see a number of collaborators there from Peter McCallum, Walter Eliza, and the Royal Melbourne Hospital. And this particular grant, as we can see there, uses a model system that we use in pancreatic cancer called organoids to identify which genes are essential for pancreatic cancer cells to survive chemotherapy. So this is very much a project around identifying potential targets that can be targeted in order to optimize the efficacy of our existing therapies for pancreatic cancer. Using, as I said, this model system, the organoid. I'll talk more about that in a moment. The reason I've flagged this grant up is because this is an example of a new collaborative grant that actually builds on earlier Accelerator Grant funding that PanKind provided quite some time ago to a major program of research. And I'll talk more about that, I think, on the next slide. No, in a couple of slides' time, so I apologize for that. Another example of a collaborative grant is this one. This is around the development of personalised treatment pathways for pancreatic cancer patients, and this is led by Dr. Sumit Sami from Colling Institute of Medical Research at the University of Sydney. Once again, we can see a significant number of collaborators there, including both local collaborators. There is an overseas collaborator who actually came from Australia, and I'll talk more about that in a moment. This particular grant is actually one where researchers have already established a significant number, or a biobank, if you like, of blood samples from pancreatic cancer patients that have been collected prior to the administration of chemotherapy. They're trying to identify unique proteins in these blood samples which can predict the response to chemotherapy. Obviously, they know now which patients responded well to chemotherapy and which patients didn't respond as well. And they hope this will eventually lead to the development of simple blood tests to identify those patients that are most likely to respond to particular treatments, and to ensure that we get optimal treatments for particular patients based on that signature. They've identified a key pathway which they believe that can be targeted to overcome resistance to chemotherapy in pancreatic cancer patients, and they're certainly looking to explore that particular pathway as well as part of this collaboration grant. The next grant that I want to talk about as a collaborative grant is a really important one. This one is called the Pancreatic Cancer Early Diagnosis Group, is the name of the group that's sort of involved in the grant. It's a collaborative. It's led by Associate Professor Daniel Crowe from Monash Health. And what they're trying to do is accelerate the uptake of precision medicine in pancreatic cancer using endoscopic ultrasound biopsy. Now, this was a 2020 collaboration grant. We can see there's a significant number of researchers involved. The reason why this grant is one that I flagged up as being particularly important is because this grant talks very much to what is happening in the global pancreatic cancer research community and also elsewhere in Australia. And it talks to many of the earlier platforms that were established in pancreatic cancer, some of which were contributed to by PanKind, but also received funding from many other sources. So this particular study, as it indicates here, is about trying to accelerate the uptake of precision medicine approaches. And it focuses on a particular biopsy procedure, which is called endoscopic ultrasound biopsy. Here we just see this is the paragraph that has been written about the summary of the particular grant, and I think I've alluded to what's on that slide already, so I'll move on to the next slide. So the vast majority of pancreatic cancers, adenocarcinomas, as we know, are diagnosed at an advanced stage, and surgery is only possible in a relatively small number, about 15 to 20%. This is one of the key issues in pancreatic cancer. The way that diagnosis is often made is with endoscopic ultrasound guided fine needle aspirates. This is the most common procedure. The pros of the procedure is it's safe, it's minimally invasive, and it can biopsy at any stage of disease. The cons of the approach are it produces small tissue samples, and that means a low yield of genetic material, and there is also potential sample contamination with stomach and duodenal cells. So this particular study tries to optimize the value that we can obtain from that biopsy. And it uses a number of resources, but the key element of the study is that the biopsy material is utilized either as a frozen sample, which is the preferred way, or from some other sort of means of storing the biopsy or storing the material. But from that, immediate work is done to obtain DNA-based information, RNA-based information, possibly the development of preclinical models. But it's really this area here that I'd like to focus on, which is the DNA-based work. And essentially what this work seeks to do is to get as much information as possible from that small amount of material using sort of a protocol that's been established. But importantly, to then try and link that information to new treatment options. Now, those treatment options are most likely to be clinical trial based, but they can also be investigator sort of initiated based and so forth. But you can see what's sort of happening here. You're basically trying to use a routine procedure that is used to try and obtain more information and to try and sort of to link that information into possible platform programs that are being undertaken. And you'll see what I mean by that in a moment. So far in that study, it's an ongoing study, and I thank Daniel for providing me with these slides for this presentation. They've obtained material from 50 patients, 44 out of 50 patients. That's about a 90% success rate. And when we say material, that means sufficient material to undertake the analyses that they wish to undertake. They got variable yields, as we can see here. There were targetable mutations were identified in 6 patients. So that's 6 out of 50, that's 12%. So that's 12% of patients when we now have information about the mutations being carried by their particular tumor, which in theory may influence potential treatment options. Of those, we can see 7% of patients had commenced on targeted therapies, and these are targeted therapies that are tied to the mutations that have been identified. And there's just a little bit of information there on the types of mutations that have been detected. Importantly, new mutations, new targeted therapies are being introduced into clinical trials all the time. So the idea is that this program will expand and more patients will eventually be presented with targeted therapy options. So their conclusion so far is that endoscopic ultrasound can be used to provide tissue for comprehensive molecular profiling of pancreatic cancer. They've been successful, as they said. And as new treatments are developed, this will become more and more important. And in particular, this point at the bottom here is that this particular study, currently largely focused in Victoria, will be expanded next year in collaboration with other studies that are being undertaken in pancreatic cancer around the country. And you'll see one in particular here called MOST-P. Daniel asked me to acknowledge the various collaborators that were involved in that project. Now, when I say that I want to contextualize into sort of a global and a national sort of framework what's happening in some of these PanKind supported projects, this is just a brief that was released by the Food and Drug Administration about 3 or 4 years ago. And it talks about the fact that we need to try and increase the efficiency of the clinical trial process, the speed to which we can introduce new treatments into the market or into sort of patients. And one of the ways to do that is to look at the way we design clinical trials and to move towards what we call adaptive trials. And adaptive trials are unique in so much as they allow design or modified design based on a established sort of protocol and established rules, but they allow changes to the design based on the data as it accumulates within the trial. This gives greater flexibility. It can be applied across all of the clinical trial phases. It certainly enables greater efficiency. And importantly, under a single master protocol, you can evaluate more than one investigational treatment, more than one disease type, more than one patient population. So the idea is that if you can set up platform trials with an established protocol, you can build from that master protocol various sub-studies that are going to help us to accelerate the introduction of new treatments. One such platform is called the Cancer Molecular Screening and Therapeutics Program, which we call MOST. This is a program that is essentially a precision medicine program that aims to link molecular targets in cancers with corresponding therapies. So by corresponding therapies, we're talking about targeted therapies that are most likely to work in patients that carry a particular mutation in a key gene. It's a novel paradigm for evaluating biomarker-driven treatments for patients with advanced cancer, with a particular focus on rare and neglected cancers. It is a pan-cancer protocol. This is if you like, the master protocol. It provides a structure for profiling tumors for actionable molecular targets, for evaluating treatments based on molecular signatures of tumors, and to permit the development and conduct of multiple parallel sub-studies of novel treatments or indications for eligible patients. Now, because pancreatic cancer is, of course, considered a neglected cancer because of its poor outcomes, many of the patients that are being recruited into this particular platform are pancreatic cancer patients. There are over 300 patients that have been recruited into this particular study. This is just a schematic showing essentially basically what happens. Patient with the doctor, lab DNA extraction. The Garvan Institute is responsible for the lab-based support for the MOST studies, and the doctor-patient discuss the results. There is the potential to involve the patient in the MOST drug trials or in the MOST immunotherapy trial or some other trial that is going to link with this particular study. Now, exciting for pancreatic cancer, as I said, there's a number of patients that have been included in the MOST study to date were pancreatic cancer patients. Next year, there will be a new study starting as a sub-study of the MOST study, which is called MOST-P. And this is a study that is supported by Cancer Institute, New South Wales Translational Program grant of 3.75 million. And it involves 2 pancreatic cancer sub-studies based on that master protocol that we've been talking about. And it will provide patients with access to either targeted therapies matched to the genomic signature of their individual tumour or targeted to the tumour environment based on signature. There are 2 trial drugs that have been developed. One's an old drug, repurposing, sulfasalazine. One is a newer drug, RxC004. Some key people involved in this MOST-P trial include Paul Timpson, and Paul Timpson was someone that we funded with an Accelerator Grant in relation to another thing that I'll talk about in a moment. Professor David Thomas, who is leading this study, Professor Marina, who is the scientific lead, and other people that were involved, Phoebe Phillips and Professor David Goldstein. And these individuals were involved in the development of the evidence base that has led to the trial drugs being ready for this particular type of study. Now, importantly, the study that I've already talked about with the endoscopic biopsy will be linking with this study. And as a result of that, it will enable the existing study, the biopsy study, to be done at other sites around Australia and to link directly, hopefully, with these clinical studies that are part of this MOST-P study. Australia is not the only country doing this type of thing, of course. The Precision Promise is one that you may wish to look up. This is a multi-centre trial to evaluate multiple regimens in metastatic pancreatic cancer. Again, it's an adaptive clinical trial platform. It aims to accelerate new treatment options. All the same things that we've already talked about with the MOST study, to create a central hub of learning, collaboration, clinical advancement, and these are things that the MOST study is doing here in Australia. And as I said, excitingly, we're seeing the growth of sub-studies in pancreatic cancer from that MOST study. Finally, I'd like to talk about the Accelerator Grants. These are the larger grants, and I just give you a couple of examples, and again, try to give you a sort of an international context, if you like. The first one I wanted to mention was a grant that was made to the Australian Pancreatic Cancer Genome Initiative. And the Pancreatic Cancer Genome Initiative, as you can see, is a global research enterprise. It has provided a wonderful understanding of the molecular basis of pancreatic cancer, and in particular allow us to distinguish different types of pancreatic cancer. Much of the work was led from here in Australia. As I said, PanKind contributed to the Australian Pancreatic Cancer Genome Initiative, and the APCGI continues to support pancreatic cancer in many ways. And again, it's really a platform technology, and studies, highly respected papers continue to emerge from this initiative and will continue. This is one of such study. This is one of the most pivotal studies, a very early one, and I just sort of flagged up 3 names on here. You can see here Lorraine Chantrell, who's a clinician on our scientific advisory panel, Sean Grimman, who was the successful applicant in one of our PanKind Accelerator Grants, and over here is, of course, the Australian Pancreatic Cancer Genome Initiative, and many of these individuals are part of that as well. Also, I want to draw your attention to something that's happening overseas, which is very similar. This is the Precision Panc platform. This is, again, a nice example of one of these platform approaches. And this has a lot in common, again, with our MOST study that we talked about here in Australia. This individual here, this is Andrew Biancan, and Andrew Biancan was one of the drivers of the development of the Australian Pancreatic Genome Initiative in the early days. Andrew was recruited by Scotland to set up a pancreatic cancer program over there. The sort of dimensions of the program are over here. It's a discovery program, a preclinical program, and a clinical development program. One critical element is, of course, this molecular analysis of cancers, of pancreatic cancers, at great depth, a very deep screening, molecular interrogation of the cancers, and then linking that to various clinical sub-studies. And I've highlighted here how their particular platform has led to a series of studies, Primaso 1, 2, 3, 4, as you can see here. And there are a whole host of new studies. You'll see that there are drug companies contributing to the funding of these various clinical studies, but they are clinical studies that link directly with a platform very similar to what's been set up here in Australia. One of our earlier accelerator grants that I just thought I'd mention very quickly also, which went to Sean Grimman, that's the name we saw just a moment ago. Sean is based in Melbourne, and Sean was successful in getting a grant to work with what we call organoids, and I've already touched on organoids. And organoids are patient-derived cancer material. We have a resection or biopsy. This is digested. There is a 3D culture using specialized methods. Basically, we support the cells with a matrix so that the cells don't attach to plates. You form a tumor organoid. This tumor organoid resembles the tumor from which it was derived. It, if you like, faithfully replicates the molecular information in that particular tumor, and you can establish an organoid biobank, which is what Sean has done. Those organoids can be used for a whole range of things, including testing for drug efficacy and toxicity. So it's an ongoing resource. And papers continue to emerge. This is a nice review that was recently written by Sean and colleagues. Tracy Putotsky is the lead author here. But it really describes all the various things we can do with organoids. We use organoids for a number of different types of cancers. We certainly use them for colorectal cancers. But much of the work in Australia was a driving force for the development of pancreatic organoids. So there's some of the things that we've funded over the years. The last thing I want to touch on were the challenges. And I thought I'd just start by highlighting one of our success stories. Marina is a highly successful pancreatic cancer researcher. She's a mid-career researcher. She's based at the Garvan Institute. Marina has received an innovation grant from AVNA in 2015, I think it was from memory. She was also a lead investigator on the grant received by the Australian Pancreatic Genome Initiative. Actually, no, it was one of the other grants around the cancer environment, looking at stroma. But Marina has been highly successful and is a sort of a star, if you like, in the pancreatic cancer research space. She's been successful in obviously winning a number of awards, and as we see across the bottom here, continued funding for her salary across her whole career. So she's been highly successful, and we obviously would like most of our key pancreatic cancer researchers to follow that trajectory. But we've already described that the success rate for NH&MRC grants is currently 10%. So what I thought I would do just very quickly is look at pancreatic cancer funded by the NH&MRC over the last 3 years of rounds. And if we look at the two major schemes, one is investigator grants. That's a people's scheme based at salary. There's only been one individual, Sean Grimman, who we've talked about a number of times, who was successful in receiving a senior investigator grant. There have only been 3 ideas grants. Shane Gray, that we talked about earlier, a grant of $925,000 this year. That obviously was seed funding with PanKind. Phoebe Phillips, who also has received some seed funding from PanKind, that received a grant in 2020. And Minotti, who was successful in receiving a Accelerator Grant, from memory 2016, 17, around then, who received a grant in 2019. That's not a lot of grants, is it, for pancreatic cancer research over 3 years? If we look at the other major source of medical research funding in Australia, the Medical Research Future Fund, a much more applied funding scheme, we see there are 2 clinical studies that have been funded through the MRFF scheme. Not putting these up to talk in a negative sense, but to talk in why supporting organisations such as PanKind are so critical. They provide that seed funding as we see. They also help to iron out the bumps in funding that researchers need to ensure they maintain their momentum. They also help to support the establishment of major platforms. And of course, those major platforms, we've seen hopefully tonight the power of those platforms. And we've seen that it is a approach that is being replicated around the world. Finally, again, congratulations to all of our researchers, and thank you to all of you for listening, and thank you, Michelle. And I apologise for my technical difficulties.
Thank you so much, Ross. Well, I mean, I think you've done an amazing job to summarise all the amazing work that's happening around the country and around the world in pancreatic cancer. My summary, not being a scientist, is that I can see that there's a number of very, very intelligent people working on a lot of different approaches to treatment of pancreatic cancer, and they all seem to be kind of attacking it from a different way, which is encouraging. I see a whole lot of collaboration between institutes, which is really encouraging, because working together, surely they can, hopefully, that results in a faster outcome. And then finally, I think trials are getting smarter and faster and more organised. So once we do start seeing some of these new treatments making some progress and showing promise, the good news is that the trials will be much more effective, so that they can reach the patients who need them much faster than ever before. So thank you so much, Ross. I'd like to conclude by saying that we could not support these researchers without the support of our community. And many of you on this call, we need to thank significantly for all the work that you have done to allow us to fund these amazing researchers. As Ross mentions, not a whole lot of government funding has gone to pancreatic cancer, mainly because it's so competitive. But I do feel confident that there is a lot of activity in the space. People are starting to talk about pancreatic cancer more than ever before. And I think that that's when we'll start to see things changing. So please feel free to come to us at any time with any questions that you might have on the research that we fund, or if you want to talk about anything. But I'd really like to conclude by thanking all those people who have helped us, and mainly today, the people who have shared their stories. Sharing people's stories with pancreatic cancer is really the way that we are starting to get this attention towards the disease. And it's such a generous thing for people to do, knowing that people are going through their own challenges, both people affected by the disease and those who have lost people. So it is with your support that we are allowed able to see this growing amount of research happening, and we're looking forward to the years ahead and the increasing support, so that we can start seeing some of this pancreatic cancer research moving forward to improve patient outcomes. So thank you for joining us tonight, and thank you so much again, Ross, for all your work, not just tonight, but for over many, many years. And we do owe you such a huge amount of gratitude. And I think the whole field owes you so much gratitude for all your efforts. So thanks again, and have a good night, everyone. See you.